Assalamualaikum and very good morning, boys and girls. Okay, so today we will learn about subtopic 3.3, which is human blood. Okay, so in this subtopic, you will be able to, first, name the method used to separate the components and constituents of human blood. Okay, the second one is to identify blood groups and the effects of receiving incompatible blood groups. Okay, meaning that what happen if we receive different types of blood groups. Okay, the third one is to communicate about the importance of blood donation in context of our daily life. Okay, so let's start. So the components and constituents of human blood. Okay, so first you have to know that our blood function is to transport oxygen and nutrients to the body cells. Okay, the second one is blood also transports waste products from the body cells. Okay, so the first one is to the body cells, okay, to be used by the body cells. The second one is from the body cells, okay, unwanted substances from our body cells. So that are the function of human blood. Okay, so can you name the component of blood which transports oxygen to all parts of the body? And then what is the color of this component? Okay, let's look at the next slide. So if you look at photograph 3.7, so this shows the components of blood. Okay, so blood is actually a type of mixture, okay, campuran. So it can be separated into two components, a yellow liquid floating on top of a red liquid. Okay, so you can refer to the figure. So the components of blood are normally separated using the centrifugal method. Okay, so photograph 3.8 shows the process of centrifugal method. So in this process, the mixture is rapidly spun. Okay, rapidly spin in a centrifuge. So you can see the difference of the appearance of blood before the centrifugation and also after the centrifugation. Okay, so before, the blood is all mixed together. Okay, all the components mixed together, but after the process you can see the mixture and then be separated into two parts, okay, yellow liquid and also red liquid. All right, so the components of blood. What are the components of human blood? Okay, so blood consists of a suspension of red blood cells, white blood cells, platelets and blood plasma. Okay, so in figure 3.12 you can see that the yellow liquid is actually blood plasma. Okay, consists 55%. And then we have white blood cells and platelets, which is less than 1%, and red blood cells with 45%. Okay, so blood plasma is made up of approximately 90% of water and 10% of dissolved substances flowing to all parts of the body. Okay, so now we are talking about this blood plasma, the yellow liquid. So it's consists of 90% of water, okay, and 10% of dissolved substances. So what are the dissolved substances? Okay, these include nutrients, carbon dioxide, enzymes, hormones and also waste products. Alright. So next one, we're going to look about human blood groups. Okay, so for human blood groups, how to determine the types of blood groups? So we have to refer the antigens on the red blood cells. So the type of antigens will determine the types of blood group. Okay, so human blood can be classified into 4 blood groups, okay, which are A, B, AB, and also O. So the classification is according to the type of antigen. Okay, if we have any of them present on the red blood cell. Alright, so the type of antigen present on red blood cell is either A antigen or B antigen, or it can be both. Alright, so look at this example. Okay, so let's say if the individual is the individual have blood group A, so the individual will have only the A antigen. So you can see that the A antigen is on the surface of red blood cell. Okay, so blood group A have only the A antigen. Okay, what about other blood groups? Okay, so this one is blood group A. Okay, for blood group B, so individuals with blood group B have only the B antigen. Okay, on the surface of the red blood cell, you can see the B antigen. Okay, for blood group AB, okay, AB, so the individuals have both A and B antigens. Okay, so you can see on the surface there are A antigen and also B antigen. And the last one is blood group O. Okay, for blood group O, the individuals do not have A or B antigens. Okay, so if you look at the figure, there are no A antigens or B antigens. So these are the 4 types of human blood groups. Okay, so how do we determine the type of blood groups? Depends on the antigens on the red blood cell. Okay, so next one is the antibodies in blood plasma. So just now we discussed about the antigens which is on the surface of red blood cell. So now we're going to look the types of antibodies in the blood plasma. So what is antibodies? Okay, blood plasma contains antibodies. So the types of antibodies present in blood plasma are anti-A and anti-B antibodies. Okay, so let's look at the table to get a better understanding. Alright, so let's say the type of blood is A. Okay, the person have the type of blood A. So the person will have A antigens on the surface of red blood cells. So the antibodies is anti-B. Okay, blood group A have A antigens and anti-B antibodies. Okay, for blood B, the person will have B antigens and anti-A antibodies. Okay, anti-A antibodies. For AB blood, okay, there are antigens A and also antigens B. So since it have both antigens, there are no antibodies in the blood plasma. Okay, meaning that there are no anti-A or anti-B. Okay, the last one is O blood. Okay, for O blood, there are no antigens on the surface of the red blood cells. So since there are no antigens, okay, the types of antibodies are anti-A antibodies and anti-B antibodies. Okay, so these are the relationship between the types of antigens and also the types of antibodies. Okay, so what is the function of knowing this information? So an antibody actually will attack its corresponding antigen and cause the coagulation of blood. Okay, so the coagulation of blood can cause death for the recipient. Okay, for the individuals who receive the types of blood, certain types of blood. Okay, for example, if anti-A, it will coagulate with A antigen, right? While anti-B will coagulate with B antigen. Okay. Alright, so these are the summary from the previous table. Okay, to make sure that you have better understanding. So let's look at the first point. If an individual has anti-A antibodies, okay, anti-A antibodies inside the blood plasma, and the person will have type B blood. So the person with type B blood cannot receive types A and AB because these two blood types contain A antigen. Alright, so we know that A antigen will coagulate with anti-A antibodies. Therefore, type B blood cannot receive types A and also AB. Okay, let's say if there's an individual who has anti-B antibodies. Okay, so if he or she have anti-B antibodies, meaning that he or she have type A blood, they cannot receive types B and also AB because we know that types B and types AB contain B antigen. So B antigen will coagulate with anti-B antibodies. Alright. An individual who has type AB blood is free to receive all types of blood. Okay, so why? Because there are no antibodies in his blood. Okay, so since there are no antibodies, meaning that there are no anti-A antibodies and anti-B antibodies, so it can receive from any types of blood. And those individuals who have AB blood is also called as universal recipient because they can receive all types of blood from any person. Alright. So on the other hand, an individual who has type O blood cannot receive any other blood type. Okay, cannot receive any other blood type. So what's the reason? Because of the presence of anti-A antibodies and also anti-B antibodies in his blood plasma. Okay, so we know that the person who has type O does not have any antigens. So since it does not have any antigens, the blood plasma contain anti-A antibodies and also anti-B antibodies. So it cannot receive the blood from A group, okay, B group and also AB group. Okay, so that's the summary from the previous table showing what's the relationship between anti-A antibodies and also the types of antigen. Okay, remember that antigen with the same antigen or the corresponding antigen will coagulate with the antibodies. Okay, for example, A antigen will coagulate with anti-A antibodies. B antigen will coagulate with anti-B antibodies. Alright. Therefore, whether a person can receive blood or not, it depends on the presence of antibodies in his blood plasma. Okay. So, compatibility of blood groups of donors and recipients. So, why we have to know the types of blood groups? Okay. So, this is important for the purpose of blood transfusion. Okay. For example, the donor's blood must be compatible with the recipient's blood. Okay, so what happens if the blood is not compatible? Is incompatible. So if an incompatible type blood is transfused, the red blood cells of the donated blood will clump together. Okay, will coagulate. So this can cause fatal blockage in the recipient's blood vessels and may cause death. Okay, so that's the effect of incompatible types of blood. So this table, okay, table 3.4 shows the compatibility of blood groups of donors and also recipient. Okay, so let's look one by one. Okay, the first one, if the donor have A blood group, Okay, so it can donate his blood. He or she can donate his blood to group recipient with blood group A and also recipient with blood group AB. Okay, if the donor is blood group B, so it can donate to the recipient with blood group B and also AB. Okay, for the person which have blood group AB, they can only donate to the recipient with the same group with them, okay, AB also. And for O, this one is very special. So the donor which have O blood group can donate to all recipients, okay, all blood types of blood group. Okay, so this one is the showing the compatibility of blood for transfusion. Okay, so what are the importance of blood donation actually? Why we have to donate our blood? Alright, so first, actually by donating blood, we could save the life of others. Okay, so for example, if we donate to the hospital, so there are sufficient blood in the hospital. So let's say if a person needs blood, they can use the blood donated by us. Okay, the second one is the blood is a living tissue that can be generated. Okay, so you don't have to worry. The blood donation does not harm the donor. Okay, so if you look here, when an individual donates blood, the total red blood cells in his body reduces. Okay, berkurang. So this forces the bone marrow to produce new cells. Okay, new fresh cells. So what happened to the individual? He or she will become more energized and able to function better because they have the body of the person have produced new cells, okay, healthier cells. Okay, the third one, blood may be needed in surgeries for treatments of accident victims as well as treating diseases, okay, like hemophilia, anemia, thalassemia and leukemia. So these are all the diseases related to the blood components. Okay. Okay, the fourth one is the donated blood can be used either as unfiltered blood for one patient, okay, or separated into components to help several patients. So we have discussed earlier, the human blood is a mixture and it can be separated into several components. So either the recipient receive the whole blood, okay, so they can also receive different components for different patients. Okay, so this table shows the main uses of each components. Okay, for example, plasma. Okay, so plasma, the main use are for patients who are suffering from great loss of blood, okay, either during surgery or childbirth. Okay, for red blood cells, the uses of red blood cells are for anemic patients, okay, which are the condition of low red blood cells or hemoglobin and causes them to feel fatigue, okay, lemah, kekurangan sel darah merah. Okay, for platelets, okay, for patients suffering from bone marrow failure and also leukemia, okay, which is the chronic disease due to the abnormal growth of red blood cells and absorb into the bone marrow. So this is for platelets. And blood proteins is for fire victims. Okay, so you can see that different components have different functions. So since the components can be separated, so one blood for one patient can help more than one person, more than one patient. Okay. Okay, so for the process of donation, okay, blood donation, we have to know the process of storage and also how to handle the blood. Okay, so look at the first one. A donor normally gives about 400 centimeter cube of blood from a vein on his arms. Okay, and then blood should be collected under aseptic conditions into a sterilized container containing an anticoagulant solution which prevents blood clotting. Okay. Next one is the donated blood will be tested first. Okay, so what is the function for the test? So we have to determine the blood group and the presence of antibodies first. Okay, because it may cause problems to the recipients. Okay, next one is screening tests are performed. Okay, to check whether donors are suffering from contagious diseases such as AIDS, hepatitis, and also other sexually transmitted diseases. Okay, so before we can donate our blood, so we have to make sure that we undergoes the test and also a screening test for diseases. Okay, besides that, the expiry date should also be written on the label attached to the blood container. So we have to refer to the expiry date. Okay, so the blood can be stored for 10 days in cold 5 degrees Celsius or longer if glucose is added. So for frozen plasma and red blood cells, they must be thawed in a water bath at temperatures not exceeding 38 degrees Celsius before being used, okay, for the patient, before being transferred or donate to the patient. Okay, so these are the components and also the storage period and the storage condition. Okay, so if we have plasma, okay, we can store it for 1 year, but it must be stored in a frozen room. Okay, for red blood cells, it can be stored, okay, 42 days, and if it is stored in a cold room. But let's say if we store the red blood cell in a frozen room, so it can stand until 10 years time. Okay, only if we store it in a frozen room. Okay, so for platelet, it can be stored for 5 days, okay, at room temperature. Okay, so we have to know the storage period and also storage condition, okay, so that we can store all the components of the blood and use it for the patients. Okay. Okay, so now look at the learning objective. Okay, the first one, name the method used to separate the components and constituents of human blood. Okay, so the centrifugal method. Okay, so this is the name of the method to separate the components of human blood. Okay, the second one, identify blood groups and the effects of receiving incompatible blood groups. So the blood groups are A, B, AB, and O. So what happen if a person receive incompatible blood groups? So it can cause coagulation of blood and also can cause death. Okay, the third one, communicate about the importance of blood donation in context of daily life. So if we donate our blood, so it can save life of others and it will not cause any harm to us, and it also can build new cells, okay, new cell for our body. Okay, so to test your understanding, so now I want you to answer formative practice 3.3, okay, question 1 until 4 and also question 5 until 7. Okay, so later we'll discuss in our group. So thank you very much for listening. Hopefully you can get all the informations that we have discussed, okay, from the types of blood, okay, the compatible blood groups, until the importance of blood donation. Okay, so Thank you very much for listening and see you in the next class. Okay, bye bye.